In the previous lecture, we talked about DNA replication in cells, and today we are going to talk about DNA amplification in the test tube. And hopefully, by the end of this lecture, it will be clear why I am making this distinction. We have already learned a lot from E. coli, and first and foremost, we have now a very good idea how E. coli assembles its replication machinery. Now the question is, can we use this knowledge in order to, let's say, make our own DNA in the test tube? Why would I want to do this? Well, if I would have the genetic information in my hands with the instructions for the function, let's say a protein, then A, I have the opportunity to make hopefully a lot of this protein, and B, also be able to hopefully manipulate this protein. Remember the cool experiment we have talked about, the quite elaborate reconstitution of the replication machinery from E. coli? Here, I need to have all these components in my hands, right? Where do I get those? Well, I can go back to E. coli and try to get the material from the cell, but recall that we have really not that much of this material in the cell. And if you wanted to really purify this machinery each time from E. coli, then you have really better chances to inflate this particular flat tire with a bicycle pump. We want to find an alternative way to make DNA, not the complicated way as E. coli does it, with this elaborate machinery that needs to be assembled each time, etc., etc., but it needs to be very simple and straightforward. So, how can we achieve this? What do we know about DNA? First and foremost, DNA is very stable, and you really do not need to be a scientist to extrapolate to that, because if you have paid attention to the news, the Neanderthal genome has now been completely sequenced, and this sequence comes from DNA material from the toe bone that is 50,000 years old, meaning if you can get the complete high quality genome sequence from DNA material that is that old, this means DNA can take a lot of punishment. If DNA is that stable, then instead of using an enzyme like in helicase in order to open up the two strands, I could simply use heat and dissolve the hydrogen bonds between the two strands, and I would have solved my problem with the DNA. But the next step would be the DNA polymerase. It needs to come in and do the synthesis. But the polymerase is a protein. And what do we know about proteins? So you know that the normal body temperature is somewhere between 36, 37. And if you see a number like this, then you better be already in the hospital because your proteins started to aggregate, degrade, etc., etc. So these two chains are not compatible. How can we solve this question? This brings us now to Yosemite with Brock and Fries. These two scientists challenged the idea that life cannot survive very hard conditions. And in order to prove their point, they made an expedition to Yellowstone, to the hot springs. And yes, they were absolutely right. In these hot springs, they have discovered a type of bacteria that they termed Thermus aquaticus that was able to survive temperatures up to 95. Not only did they discover this particular organism, they also have been able to culture this bacteria in the laboratory. And this discovery has changed the course of molecular biology later on. And just out of curiosity, I think right now the record is with M. candleri. This looks like a bacterium, but this is actually an archaea. And this one can go up to 120, and this is again very impressive. Now we have discovered an organism that has to have a DNA polymerase that is stable at practically boiling temperatures. So now we have the inherently stable DNA and now also heat stable DNA polymerase. And this together brought us the revolution in biotechnology, the polymerase chain reaction. Work of many brilliant people eventually established a method in order to make DNA in high quantities outside of the cell. And the beauty here is that this method is exceedingly simple. We only need few gradients. So we of course need the DNA template where the information is on. We need the primers, we need the 4 letters, and of course our heat stable DNA polymerase. When we have all these things in the test tube, the only thing what we need is practically an oven that can heat up and cool down. So we heat in order to separate the double-stranded DNA, cool a little bit down in order to get the primers hybridized. We have all 4 letters in the soup, and the DNA polymerase can simply start synthesizing the new strand. The steps we have just discussed, these will be then repeated over and over and over again until we have enough DNA material in our tube. In the next slide, we will see a visualization of the polymerase chain reaction, and there you will appreciate how efficient this method is in order to amplify DNA.
The polymerase chain reaction, or PCR, amplifies a specific DNA fragment from a complex mixture. First, the mixture is heated to separate the DNA strands. 2 different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the desired fragment. After lowering the temperature, the primers hybridize to the DNA, where they bind specifically to the ends of the desired target sequence. A heat-stable DNA polymerase and nucleotide triphosphates are added. The polymerase extends the primers and synthesizes new complementary DNA strands. At the end of this first cycle, 2 double-stranded DNA molecules are produced that contain the target sequence. This cycle of events is repeated. The mixture is again heated to melt the double-stranded DNA. The primers are hybridized, and the DNA polymerase synthesizes new complementary strands. At the end of the second cycle, 4 double-stranded DNA molecules are produced that contain the target sequence. In the third cycle, the mixture is heated, the primers are hybridized, and DNA polymerase synthesizes new complementary strands. At the end of the third cycle, 8 double-stranded DNA molecules are produced that contain the target sequence. 2 of these molecules are precisely the length of the target sequence. In future cycles, this population increases exponentially. Cycle 4. Heating, hybridization, DNA synthesis. At the end of the 5th cycle, there are 22 double-stranded DNA fragments of the correct length and 10 longer ones. Cycle 6, 10, 15, 20. After 30 cycles, there are over 1 billion fragments of the correct length, but only 60 longer ones. The product, therefore, consists of essentially pure target sequence.
let's go back to our title slide where we made this distinction between the DNA amplification in the test tube and DNA replication in the cell. So here we see again the PCR steps, but here we also see the 5' 3' directionality of the parental strands. So top we have 5 to 3', and bottom of course the complementary 3 to 5'. The primers anneal, and now the DNA polymerase can come in and extend the strand. But we see that top panel, the synthesis goes to the left, and bottom panel, it goes to the right. And this is the fundamental difference between a DNA amplification in a test tube and DNA replication in the cell. The machinery that works in the cell is unidirectional. So the synthesis direction is the same for the top and bottom strand, if you will. And here we have a purified polymerase in our test tube, and it is not confined in the machinery and doesn't do this physiological replication. And this is the major difference between the amplification in the test tube and replication in the cell. Now we have our target gene amplified in our hands. Now the question is whether or not we can introduce this gene into E. coli and convince it to also make the product for us. Remember, E. coli has a circular DNA, and one might think that we should introduce our gene in between so it also gets done, but this isn't how it's done. Instead, it's going to be a lot simpler. We are going to create our own circular DNA that is much, much simpler. Remember, we already learned one word in genomics, origin of replication. In order to convince the machinery in E. coli to also recognize our vector, we basically introduce this word into our vector. Here we see a typical cloning vector, the so-called, and the first thing that we realize is that this vector has about 2,500 base pairs. It can range between 2,500 up to 10,000 base pairs. But at the end, compared to the circular DNA, the genomic DNA of E. coli, this is all extremely tiny. And we already learned that we can put this word onto this vector, origin of replication, so that the replication machinery definitely also recognizes our vector and does the synthesis. But the other question is, E. coli will swallow DNA material. This is what they do, but they also spit it out if they don't need it. The question is, how do we convince E. coli to also keep our vector all the way down? So what we learned the previous lesson is that we can actually pretty efficiently kill bacteria because we sort of figured out how it works. So what about putting an antibiotic resistance on the plasmid and making sure that E. coli does not spit it out when we add this antibiotic into the medium in which E. coli has to grow? If it spits it out, it doesn't have resistance, it will die, so it will definitely keep it. So up to now, we have prepared our vector, made sure that the vector gets recognized by the replication machinery of the E. coli. We made sure that when E. coli swallows our vector, doesn't spit it out again, but we still have a big problem. We have our PCR product amplified in our test tube. The question is how we are going to get it inserted into this vector. This is again a non-trivial task, and we are going to have to learn from nature again. And this is going to be directly from E. coli because it makes enzymes called endonucleases that can introduce specific cuts. And these are words that these enzymes can recognize, and these words are palindromic. When you look, for instance, at the EcoR1 that is circled in black here, you will read GAATTC on top, and at the bottom it goes back GAATTC. So when the enzyme sees such a sequence, it will introduce a cut. So here we see a crystal structure of this enzyme wrapped around the double-stranded DNA, shown here in pink and green. So it will basically scan all the way through, back and forth, until it sees such a sequence and then introduces this specific cut. And you see, we have now sticky ends, if you will, that we can then anneal later on. And this is how we can then make compatible ends both on our PCR product that we want to put into the vector, and of course also in the vector itself. So this would basically solve our problem. But the other question is, of course, why would actually E. coli make such an enzyme that can theoretically also cut its own DNA? Well, it has a very good solution for that. But first, the reason. E. coli also has enemies. Recall, we talked about the bacteriophage that destroys the E. coli by injecting its DNA material. And when DNA or foreign DNA enters the E. coli, these enzymes basically scan through, find these sequences, and destroy the viral DNA. Now, how could it be that the own genome is protected? Well, it basically marks its own DNA. In the case of EcoR1, for instance, it would be the GAA position. The second A would get a methyl group, and the enzyme, when it sees this methyl group, would then know, that's my genome. That's the one that I'm not allowed to cut. So that's, again, a very neat trick from nature right here. Today, we have many of these enzymes that have been discovered over the past years, and all these enzymes would then recognize these small words, and all these words, when cut, would introduce these compatible ends. And what do? We go back to our vector and put bunch of these words in a specific position that is called multiple cloning site, so that we can then pick and choose what we want to combine in order to create, and this is important, 2 different ends, so that we can put in our gene of interest unidirectionally. Why this is important, we are going to see later. With that, we are already also at the end of our cloning process. So when I make my PCR product with the compatible ends, the only step that is left is to basically join the PCR product with my vector. And for that, the enzyme we are going to use is the DNA ligase. Recall, this is the machine that we need during the replication in order to join these little Okazaki fragments during the DNA replication in E. coli. And you see, we keep using all the enzymes from E. coli in order to make our own recombinant DNA. So basically, we learn from nature and use it to our advantage. And lastly, in order to have good quantities of this vector, we go back to our bacteria. Remember, we have the antibiotic resistance on our vector. So the simple trick, we grow the bacteria in the medium that has this antibiotics. So the E. coli will definitely not spit out our DNA. And we just grow the cells and purify our vectors. At the end of the day, we have practically purified a gene, and we have been able to do so because we understood the replication machinery in E. coli, so we learned a lot from nature. Plus, we also have learned to speak a little bit of genomics, so we started to understand a couple words. And when you put all these things together, this was basically the beginning of modern biotechnology. And this is really not that bad for a 4-letter alphabet. So far, we talked about making DNA, either in the cell or in the test tube. Next, we want to understand how the information on the DNA gets extracted. And as always, if you have any questions to part 5, please drop me a line.